And we will wait probably another minute or so before we get into it. But as I said, there's a few announcements for today, so let's get started on them. So the presentation today will be an update from the Pharmacogenomics Working Group. But before we get into that, firstly, I want to take this opportunity to again congratulate all of the individuals who are recognised as part of the second annual ClinGen Excellence Awards. So there's a link to the webpage about that there. We'll put that in the chat. So you can read more about all the individuals and their contributions that they've made that we felt was worthy of recognition. But I do also want to take this opportunity to thank all of the contributors to ClinGen. We really couldn't do the work we do and achieve everything that ClinGen has over the last 10 or 11 years without the contributions of our volunteers. So this is an opportunity to recognise some of those individuals who have gone above and beyond, but we do also greatly value the contributions of everyone who volunteers for ClinGen. I also want to mention that over the next 5 or 6 weeks, we have a ClinGen Clinical Genomics Careers panel series. So again, the link for that is displayed there. We'll put that in the chat too. So it's a different theme every week. I think coming up next week is about clinical laboratory careers.
Can I get something? No, we'll touch it too.
Encourage people to check that out. And we will be adjourning the Biocurator workgroup meetings for the summer. So this will be our last meeting for the next few months, and we'll reconvene on the 11th of September. So keep an eye out for those cancellations coming through. I know sometimes Google Calendar, which we use, doesn't always play nicely with Microsoft, so some people might need to manually delete their meeting holds for those, but they should be coming through. But with that, that's all the announcements sort of from an administrative perspective. I'm not sure if anyone else on the call has announcements that might be relevant to this general forum. But if not, we can get into the presentation for today. So as I said, it's an update from the Pharmacogenomics Working Group, and with us today we have Lee and Clarissa, the coordinators of that working group, and also Michelle, who is one of the co-chairs of that group. So with that, I will hand over to them.
Great, thank you. Lee, are you going to do the outline or? Well, I think she's muted.
Sorry,
there you go.
Yeah, just go ahead, Michelle. Yeah.
Okay. Okay. So we divided this talk up into a little bit about the background of pharmacogenomics, what it is, why we care about it, and what kind of makes it different from medical genetics. And then Lee will talk about actual curation for pharmacogenomics. And then Clarissa is going to talk about more about the working group specifically. And I'm going to add on a little bit about what our work to date has been in that group. So thanks, Lee. So yep, so first section is me. Why do we care about pharmacogenomics? But what is it? So pharmacogenomics or pharmacogenetics, the name is pretty self-explanatory, pharmacology and genetics or pharmacology and genomics. And basically, it's just the study of how an individual's genetic makeup affects their response to the medication that they're taking. And the whole goal of pharmacogenomics is to have a better efficacy, have a medication that works better for people, and lower toxicity or less side effects for people. So it is personalized medicine in that our goal is to have the right dose of the right medication for a person at the right time. The clinical utility of pharmacogenomics has been established. There are definitely working guidelines about how a clinician can prescribe medication using genetic information. FDA has some of this information on some drug labels as well. And so we would really like to integrate pharmacogenomics into the overall, medical genomics area in order to together have a more holistic goal, or achieve the goal of personalized or precision medicine. So some of the differences between pharmacogenomics and medical genetics, or more disease genetics. So they're similar in that they both have to do with genetics, but of course, pharmacogenomics, as I said, is focused on drug response, so and optimization of therapy. So the nomenclature and the terminology used between the two fields differs a little bit in that in pharmacogenomics, we're very concerned with the pharmacokinetics and pharmacodynamics of drugs. And we have different nomenclature for variation because many times metabolizing enzymes, which are very important for drug response, and the genes encoding these metabolizing enzymes have a lot of variability in them. Oftentimes, it's a haplotype across the entire gene that's important instead of individual variants, and those haplotypes are called star alleles. And I'm going to go into that in a couple minutes in a little bit more detail. But, generally speaking, a person that's walking around with a genetic variant in these pharmacogenes is perfectly, you would never know. There's no effect on them until they take a medication that is affected by those variants. So completely different in that sense than medical genetics, because you don't really have a phenotype until there's medication exposure for the most part. And let's see, what else? Is there another big, huge difference here? I don't think so. I think that's the primary difference. Oh, and one thing though I did want to say is the frequency can be different. So many times in medical genetics, you're looking for something at very low frequency because if people had this, this variant at high frequency, it would probably be, uh, those people wouldn't be reproducing. So the frequency goes down. That's not the case for a lot of the genetic variation in pharmacogenes that can be quite prevalent because, as I said, there is no phenotype until the medication exposure. Sorry, go ahead, Lee. Okay, so the field of pharmacogenetics has been around for several decades now, and in the last 20 to 25 years, there's been a real push to have online resources supporting the field. And so these are some of the biggest sources of information outside of just peer-reviewed published literature in pharmacogenomics. First, we have the FDA has some information on their drug labels about the impact of genetic variation, and they have a couple tables that they put out that summarizes some gene drug associations. The next resource is PharmVar, which is a nomenclature resource which defines those star alleles or those haplotypes across the pharmacogenes that I just mentioned. We have CPIC and the Dutch Pharmacogenetics Working Group next. These groups write guidelines for clinicians about if you have some of the variants defined in PharmVar, what specifically can you do with prescribing a medication based on those variants? And lastly, we have PharmGKB, which is an overall pharmacogenomics resource and actually contains information from all of these other resources on the slide, plus much more. So everybody on the call, Lee and Clarissa and myself are involved in PharmGKB and CPIC efforts. So those is kind of overlapping group responsible for those two efforts. And together, we're looking forward into the future of kind of combining these groups together along with some, yeah, go ahead, Lee, thanks. Along with some other projects that we work on. We have a software tool, et cetera. But the goal is kind of to take some of these kind of disparate resources and put them together into a resource that will ultimately be called ClinPGX. That's a proposal right now. And the proposal is for ClinPGX to kind of be like a sister organization to ClinGen. ClinPGX could cover pharmacogenomics and interact with ClinGen and submit to ClinVar. And be part of this overall ecosystem of genomic medicine and be responsible for the genomics piece. And so pharmacogenomics piece. And so ClinPGX would contain maybe tools and so forth for curating pharmacogenomics knowledge. Now, PharmGKB already does some curation of pharmacogenomics knowledge, and Lee's going to go into that in a minute. But as we move forward, I'm going to be talking about tool development later on that will be under the umbrella of ClinPGX. Next slide. Yeah, so as I said, if ClinPGX's goal is to kind of be part of like a sister organization almost to ClinGen and be very aligned with it and part of the same ecosystem, our intention is to develop kind of parallel frameworks for curating pharmacogenomics. Now, as I mentioned, these resources have existed for 25 years. There is standardized terminology and ways of curating pharmacogenomics information currently. And like I said, we're even putting out guidelines for how clinicians can use this. But it has been developed kind of independently from what's been developed in ClinGen for medical genomics. And we know that ClinGen is really the gold standard right now in genomic medicine for these kinds of frameworks for defining validity and actionability, et cetera. So one of the goals of the PGX working group is to come up with parallel frameworks to ClinGen and ultimately implement them under the umbrella of ClinPGX. Okay, next slide. Okay, so just quickly, I mentioned that there is a difference with nomenclature and variation. You can't be involved in pharmacogenomics without dealing with these things called star alleles. They're completely different than anybody has seen before if you're on the medical genomics side, and there's a lot of resistance to them, and I understand why. They're strange things, but they've been around for decades now, and I don't think it's going anywhere. They also have a purpose. So I'm going to show you that in just a second. So here we have an example of a genetic variation in the CYP2D6 gene. So CYP2D6 here is the gene name, and star 8, maybe advance a couple, yeah, and star 8 is the allele designation. And so what we mean by allele here, when we talk about star alleles, is a haplotype across the entire gene. So this star 8 is actually, if you go the next advanced one, yeah. So here's the HGVS representation for star 8. It's actually 3 different SNPs across the gene. That's really difficult to write out and to say. It's much simpler to say star 8 or write out star 8. So that's why it's kind of caught on. But it has kind of a greater implication than that. So not only does it mean these 3 variants, it means only these 3 variants. So it means like we kind of have an idea of what is going on across that entire gene of CYP2D6, and we're only seeing these 3 variants. If you only see these 3 together in this combination, that is what we're referring to as a star 8. So in these star allele designations, you can have SNPs, indels, repeats, there are structural variants, sometimes copy number variants. Importantly, in pharmacogenes, some very impactful variation can be outside of the exomes. We can have intronic variation that leads to, they're like affecting splice sites and so forth, or upstream or downstream of the gene, then affects transcription of the gene or some kind of regulation of the gene. And right, so this star allele has a function that's assigned to it, and it's the combination of the variants across that entire gene that are responsible for the allele function. And I think I mentioned before that the allele frequency might be quite common for these. So next slide. So, right, so it's the 2 alleles together that, this is not, this is not unusual for genetics. It's the 2 alleles together that give you the overall phenotype in the patient. So if a patient had 2 quote unquote normally functioning copies of the allele, they're considered a normal metabolizer. If, however, one copy of the gene on one of the chromosomes is affected so that it doesn't function as well, it has very little or no function, then overall that person is only has activity enzyme activity about 50% of the first person that has no variant alleles, and they're called an intermediate metabolizer. If both alleles are affected, they'll be called a poor metabolizer. And there are cases where you can have someone can create be expressing even more protein, or the protein's even more functional than the person having 2 normal function variants, and they would be called a rapid or an ultra rapid metabolizer, depending on the gene. And just to give you an example of how this works in the real world, on the next slide, we're going to talk about clopidogrel and a gene called CYP2C19. So clopidogrel or Plavix is an antiplatelet. It's given to people who have had a cardiovascular event, like had a heart attack or something. We don't want them to clot more. We want the blood to keep flowing. So this antiplatelet, makes the blood less sticky, if that's, And so this drug in and of itself, when it's taken by the patient, doesn't have an effect. It has to be metabolized into the active metabolite for it to have its antiplatelet effectiveness. So CYP2C19 is the enzyme that does this. We also know CYP2C19 is a lot of variation across the gene that can cause to have poor function or increased function. And so the patient's phenotype is dependent on those two alleles and being exposed to the drug. And so on the next slide, we talk about, again, 2 copies of normal functioning protein. They're a normal metabolizer. Drug works as it should. If they have 1 copy of a protein or 1 copy of a gene that has some kind of functional impact variant, they're only metabolizing half of the clopidogrel they take. They have less active metabolite, decreased efficacy. They're at risk for having an adverse event because the medication is not working well for them. And if they have 2 impacted alleles, it's almost as if they're not taking any medication at all. Did I freeze? Yeah, it's almost as if they're not taking any medication at all. And so they're at risk of having an adverse event. And it's actually not the drug causing the adverse event in this case, right? It's the fact that they can't metabolize the drug. So the drug is not even, it's as if they're not taking it at all. So that's why we care about things like that. Those people, poor metabolizers, they really should be taking a different drug altogether. Putting them on clopidogrel does nothing for them. Next slide. Right, so like I said, it matters the variance because that impacts your diplotype, which impacts your phenotype, and that is going to impact, the prescribing recommendations, like if someone should take a different drug or have a different dose of medication. Okay, thanks. I hope that sets the stage, and I'm going to turn it over to Lee.
All right, thank you, Michelle. So I'm going to give a brief introduction on how we annotate pharmacogenomics information at PharmGKB. So, why we talk about this? We think this really would be helpful to understand why we develop, relate to how we develop clinical validity and actionability for the pharmacogenomics working group. Why we leverage the existing knowledge resources to build the framework. So PharmGKB is a centralized knowledge resource that curates pharmacogenomics information. We're housed at Stanford. We're NIH-funded resource, free online, and we're hoping to be a one-stop shop for pharmacogenomics information. And here's our website, PharmGKB, www.build.pharmgkb.org. So if you haven't tried it before, I really encourage you to go check it out. And the next slide shows the 3 primary sources of knowledge we have at PharmGKB. This includes peer-reviewed published studies. We also have information from the FDA. This includes, as Michelle mentioned earlier, we have information from the FDA drug labels. FDA also have a resource posted 2 years ago. This is a table of pharmacogenetic associations. And the third type of knowledge we have at PharmGKB, we curate at PharmGKB, published clinical dosing guidelines. These are guidelines on how to adjust treatment of certain medication based on a person's genetic medication. And so I'm going to go into more detail on the 3 types of knowledge listed here. So first, let's talk about one of the primary source of knowledge we have at PharmGKB is peer-reviewed published studies. And as compared to medical genetics, so evidence for pharmacogenetics variants comes from association studies rather than case levels data or segregation data. So the primary knowledge, primary source of evidence comes from peer-reviewed publications. And at PharmGKB, we use both manual as well as automated method to curate literatures on pharmacogenomics. As you can see from this slide on the bottom half here, so this shows that we use we have one way to get pharmacogenomics annotation is through manual routine journal scans. So we scan about 12 different top pharmacogenomics journals. Our curators does that, and to identify relevant articles that we can curate for varying drug relationships. So in addition to manual curation, we also have automated method. So we use 2 main automated methods. One is called lead suggest. Many of you probably are familiar these are tools from NCBI, and we also have another machine learning tool called PGX-Mine, which are specifically developed to extract pharmacogenomics relevant gene-drug associations from articles. So both manual as well as automated method were used to identify pharmacogenomics articles, and the articles. So we get articles weekly, and then it will be triaged to our curators. The curators will then write detailed variant drug summaries, which in a very structured way and can be stored in PharmGKB knowledge base. So here just shows you, one of the examples of the papers that we have at PharmGKB. So a curator will take a study like this. This is a landmark study published in New England Journal of Medicine back in 2008. So this is a discovery paper that the group finds functional variants in SLCO1B1 and how that's associated with statin-induced myopathy. And so we will take a study like this, and the PharmGKB curators will then extract key information from the paper. For example, which variant alleles are mentioned in the study, and what is the direction of association? Meaning, like, is this an increase? The variant is the variant associated with increased response to a medication or decreased response of the medication? And we also capture statistics mentioned in the study, for example, p-values, as well as the cohort size from the study. And then we annotate the paper with standardized terminology. So we use HGNC nomenclature for genes, and we also have a drug ontology, and we use a standardized phenotype ontology as well. So here just shows you a brief example of our variant annotation. Can see here that our curator will write, we use a online tool to write structured summaries like this. So genotype CC is associated with increased likelihood of muscle diseases when treated with the drug here, simvastatin. And normally we write it in a way that we compare one genotype associated with a specific phenotype compared with another genotype combination. So each of this variant annotation will be tagged with gene, variant, and specific phenotype categories. And for example, this paper discussed toxicity for a variant drug relationship. And we also capture detailed study parameters, for example, the study type, study size, and associated p-value and their statistical significance, as well as which population are mentioned in the study. So we take many studies like the one I just showed you previously, right? Variant annotations, and we group together, all the variant and all the papers that mention the same variant drug phenotype combinations, and we summarize them into a genotype level clinical genotype level summaries, how this variants and what is their clinical impact on drug response phenotype, which we call clinical annotation. And each of this clinical annotation will be scored using a semi-quantitative method. I'll show you, I'll explain more later in the talk. And in addition to the published literatures, we also curate clinical dosing guidelines published by professional group. And Michelle mentioned like the two primary group of clinical dosing guidelines. One is called CPIC, which is based in US, and another one is called Dutch Pharmacogenomics Working Group, which is based in Europe. Those are the two main clinical dosing guideline development groups that we work with. And here at PharmGKB, you can find all the annotations for the major pharmacogenomic clinical dosing guidelines. This is a landing page for the dosing guideline annotation. We normally include the summary statistical graph for the data source, showing you about how many guidelines from each resource. And we also use tags this tags we use across PharmGKB database to indicate whether this annotation mention alternative drug should be used for a specific genotype or phenotype, whether it has dosing information, or if there's some additional clinical guidance given based on a specific genotype. And so here you can see, so if you click on a specific clinical guideline annotation, this is one example annotation page. It's a CPIC guideline for citalopram and CYP2C19. Citalopram is a commonly used medication to treat depression, and variants in the CYP2C19 gene has been related to responses to that citalopram response. So we annotate these clinical guidelines and provide an interactive dosing table in the middle of the guideline annotation page. So for example, here, you can use this pull-down table to select your diplotypes or genotype combinations. And then, after you enter the genotype combinations, they will show you the specific phenotype, phenotype or metabolizer status for this diplotype combinations, as well as the specific clinical recommendations for this diplotype. So for example, here, CYP2C19 STAR2, the CPIC recommendation is they will consider 50% reduction of the starting dose for citalopram. And last but not least, one of the main sources of, very useful sources of pharmacogenomics knowledge also comes from the drug labels. And we use both the FDA-approved drug label information as well as the table of Pharmacogenomics Association. And PharmGKB curate not only labels from the US, we also cover a number of drug labels from regulatory agencies across the world. So for example, from the European Medicine Agency, as well as regulatory agencies from Canada, as well as Japan and Swiss. And the most recent collaboration we have is with a group in Spain. We're going to annotate labels coming from drug labels from Spain. And I also want to mention that, we don't annotate all the drug labels. We only annotate drug labels with gene or information contained within labels. And at PharmGKB, because sometimes the language in the drug label are very vague, and it's very hard to interpret, what are the relevant pharmacogenomic information contained with this label. So PharmGKB developed, set up a framework for assigning different levels of PGX information contained in drug labels. This category, are very well adopted by many people when they mention PGX information in drug labels. So we separated currently into 5 different categories. We use them to tag drug labels to indicate whether genetic testing is required or recommended, as stated in the text in the drug labels, or the language in the label only include things that they're informative, but the FDA didn't, but the drug label didn't say whether any testing is required or recommended. And we also use a specific tags to highlight PGX information on the table. More specifically, we have 3 different tags here. The dosing, if the label has a dosing information tag, that means that the label mentioned a dosing adjustment is recommended for a specific genotype or phenotype. And if you have this alternate drug tags, that means, it's the label has specific language mentioning that a different medication should be used for a particular, for a subgroup of patients who carry a particular genotype or phenotype. And we use other guidance tag to indicate whether there's other language besides dosing change or use a different drug. For example, does it require, does the label mention, doing additional monitoring, clinical monitoring for this, for a subgroup of patients carry a specific genotype or phenotype. And I included a link here at the bottom if you want to learn more for how we define different levels of PGX information in the drug labels. And here shows you the landing page that we have for FDA labels. Using this table, you can filter drug labels to see whether the FDA PGX association table mentioned this gene drug combination. Is it at the recommendation level, or it mentions safety or response? And you can filter by whether it's only pharmacokinetics only. And you can filter or sort the table by their PGX level of evidence, as well as the information tags we have here on the right-hand side. And this just shows you an example FDA label annotation for abacavir and HLA-B. And you can see we will mention which variants are mentioned in the label. And we have a very high level summary written here, summarize the PGX content in the label. And just to we talk about there's three main type of knowledge that we curate at PharmGKB: annotations from the literature, the FDA label annotations, as well as from the guideline. All of this information we bring together to write this genotype or allele level drug phenotype summaries, which we call clinical annotation at PharmGKB. Here shows you the example page for clinical annotation for this variant, this SLCO1B1 variant, and how it's related to statin-related myopathy. So, by providing this summary type of information, so the user wouldn't have to go into to do their own search in the PubMed to look for information related to this specific variant drug combination. And so we provide the summary text for each genotype for this variant gene, variant drug combination. And so here I'm just giving you more detail on how we do our clinical annotations. Each clinical annotation is assigned a level of evidence, which is based on many different factors, including whether its implementation status, whether that means whether it's mentioned by the clinical dosing guidelines or is mentioned in the drug label. The levels also depend on the statistics from the study, whether it has been replicated, what is the type of study, and the population size. All of this are taken into account to assign a score for PharmGKB variant annotations, which then group together. We sum up the scores, and the summary score will be used to assign a level of evidence for the clinical annotation. So currently we have four different levels of clinical annotation, from level 1 to level 4, and with level 1 being the highest for associations supported by clinical dosing guidelines and drug labels. And I also put the links here on the slide if you want to find more about how we assign level of evidence for gene-drug relationships, and you can go to the links here. And so this is, a rough overview of how we curate knowledge at PharmGKB. And I'm going to turn it over to Clarissa, who will share more information on the pharmacogenomics working group and our goals and activities. Clarissa?
Yep. Thanks, Lee. All right, so the pharmacogenomics working group under ClinGen, you can see the web page here. Lee and I both coordinate. Michelle is one of the chairs alongside Terry Klein and Stuart Scott. And like Michelle kind of articulated, our goal really is here to bring, pharmacogenomics into kind of alignment with what ClinGen in that space is doing, using a couple different, approaches to this. First, thinking about kind of aligning terminology, and then really kind of incorporating PGX knowledge into, how ClinGen does things. So you go to the next slide. We're really lucky to have broad membership from a variety of fields, including different facets of what makes pharmacogenomics important, people who specialize in that, people who specialize in pharmacology, molecular diagnostics, medical geneticists, regulatory, and more. So you can see some of our members here. And then, yeah, so the purpose of the working group, we have a couple of different things, but the real goal is to kind of build out these frameworks that Michelle mentioned. And so what we want to do is we want to build frameworks for both, gene drug clinical validity and gene drug clinical actionability to align with the existing ClinGen frameworks. As a piece of that too, we want to think through how we do variant classification, as is the ACMG criteria, for pathogenic to benign classification. It doesn't really work with PGX for reasons, in part being that you don't really have kind of a pathogenic PGX variant because typically it's related to taking actual medication, right? As Michelle said, a non-functional variant, allele, it doesn't necessarily mean the patient is actively at risk for lack of efficacy or toxicity. It completely depends on both, what the second allele is and also the drug and the drug class. So it's a bit more complicated than just straight Mendelian genetics. And so, by building kind of consistent terminologies and frameworks that are applicable to PGX alleles, but that also kind of make sense with and are, aligned with what the clinical genetics community is currently using, the overall goal is to make PGX implementation easier and support it better, right? So to reduce reporting inconsistencies between different labs who are using their own nomenclature or pulling from different places or trying to figure out what they're doing, to also help, if we have a kind of a gold standard that's works, works with what we already have on the ClinGen side, that would really help too with labs thinking through, okay, which genes do we actually want to test for? What's valuable to test for? What's a good way to determine which genes are valuable to test for? And so, the overall kind of process we have for this is really thinking through, okay, here's criteria for how we want to evaluate PGX relationships. Looking at both this, clinical and then also functional evidence we have in, the literature, existing databases. Here's how we want to leverage existing efforts from existing resources. So we want, buy-in from the broader community. And so here kind of you can see an overview of what our current major activities are. As you said earlier, we're really thinking through the validity and actionability frameworks point. Validity here, we're talking about, looking at the evidence to determine if there is an association between, variation in gene and a particular drug response phenotype. Actionability is a little bit different in PGX than it is in kind of clinical genetics. Here, this is really thinking through looking at the evidence to determine whether or not we can actually use this variation to guide prescribing, whether that means, there is enough of a change or there's actually something we can do, an alternate drug, can we adjust the dose practically, does that actually work? And then bringing those two into alignment with the ClinGen frameworks, as well as leveraging existing efforts so we're not kind of recreating the wheel from scratch. Including all of those wonderful resources that Lee and Michelle mentioned earlier, leveraging the curation efforts from the PharmGKB and those strategies, as well as the guidance from CPIC, the DBWG, what's existing on the FDA labels, and the other ones we curate. And so, yeah. So as part of this, or kind of a precursor to this, one of the things we did was really wanted to do some needs assessment surveys. We conducted 2 rounds of these. The first one was really geared towards the PGX community, people who spent a lot of their time in that space. The second one was geared towards the broader clinical genetics community, wider stakeholders, including people who didn't take the first one within the PGX community. Thank you to everybody who is on this call who ended up with the survey and took it. We really appreciate it. And we were able to get kind of pretty broad participation from both communities, 508 people after the inclusion criteria. We have, more before that as well. And, from this, what's really useful, I think, discussing on this call is there was really kind of broad support from the respondents for standardization under one, single PGX specific resource, which is exciting to see, as well as additional support from kind of both groups of stakeholders for leveraging ClinGen frameworks that existed to develop these PGX validity and actionability frameworks, as well as a lot of really useful feedback and free text regarding how specifically to do that. I just also want to note that of those numbers kind of presented on that second to last bullet point, those are the people who said yes, leverage with, commentary perhaps, but no, restrictions. There's a significant portion of people who said no, who said in their free text responses, not as is, but you have to account for these other things. And then, yes, I think we should leverage them as well. So it's, that was really exciting to see and kind of validating the approach we're taking. The manuscript is currently in preparation for this as well. So look forward to seeing that later. And then if you, yeah. And so, let's see, my screen is vanished for some reason. Yeah, and so then the process for developing the PGX frameworks, I think Michelle will also chime in here a little bit. So, feel free whenever. But, one of the, first approaches we had was we really want to get both of those pieces together, right? The existing PGX standards, the approaches, what people are doing, and then kind of taking the community input from the surveys as well towards those frameworks. And so, Michelle, I think you want to talk a little bit about those.
Yeah, like Clarissa said, using the input from the surveys, the existing standards, and what's going on in ClinGen. So we're very fortunate to have members of the PGX working group from the ClinGen validity and actionability groups who presented to us and help us align there. And we're just taking all that information together and coming up with a parallel frameworks for pharmacogenomics, drafting the definitions, creating the score assertion and scoring matrices, and then just testing it out with gene drug pairs and iterating on that until we get where we want to be with the scoring. Next slide. Right. So, this should look familiar. It's the same levels of evidence for the gene disease validity. We are aligning with that. So we're using the same terms and the same levels, changing the definitions a little bit slightly so that it makes sense with the evidence that we have for pharmacogenomics. But that's the goal there. And next slide. So then I'm not going to show you the definitions per se, but, in parallel with how it's done with gene disease validity, just coming up with scoring ranges for each one of these evidence levels. Is specifically strong through no known PGX relationship. And then the expert panel that reviews at the end would also weigh in on whether or not something that is strong has been repeated over time enough so that it would meet the definition of definitive, or on the other end of the spectrum, there's enough kind of counter evidence or evidence that would maybe dispute or refute a particular association. And these are some examples of gene drug pairs that we already know based on the drug labels, the guidelines, and the peer-reviewed literature that we already curate where we would expect them to be. We mentioned that actionability is a little different from validity, but of course it makes sense. It's only logical that in order for a gene drug pair to have some kind of actionability, that they have to be valid. So we wouldn't consider a gene drug pair for actionability unless it scored a moderate or above over the gene drug validity. Next slide. This is just a kind of overview of our workflow. We collect evidence from these existing sources or the peer-reviewed literature if there's not anything that's already like on an FDA label or in a guideline. Evaluating the evidence based on clinical outcomes, the pharmacokinetics of the drug, if it is a pharmacokinetic association, and experimental data. And our experimental data also involves pharmacodynamic data, if that exists. Assigning the rating, and then the expert review group would come in and look over what was done and have the ability to change if there is expert consensus to change a rating or to push a rating into the definitive or the disputed or refuted categories and before finalizing for posting. This is an example of our framework sort of aligning a little bit with the gene disease stuff. So you can see on the left-hand side, our big outcomes are clinical outcomes, pharmacokinetics, and the experimental data. And then broken down from there, we would have input from the different existing resources, score based on that. We have caps on how many points an association can score in any particular category, then summing that all together to get a proposed final score. Next slide. So this is how we would get to the scores that go into that previous matrix. Again, we have you've heard it many times now, these groups, evidence from the guidelines, from the drug label, or from the literature. So in the second category there, the level, those levels are existing levels for those types of evidence. So for example, CPIC already rates its evidence for pharmacogenomic associations from high to no association, versus Dutch group uses a numeric scoring system. FDA uses some different language in their tables. PharmGKB has its own system of level of evidence that Lee went over. So because they're all using kind of different things, we're harmonizing it here. We're trying mapping to our gene drug validity score using our framework. So, so that you can kind of equate a high level of CPIC evidence to a score of 4 in the Dutch working group. So these are the proposed scorings. We're still working on finalizing this, but this is our proposal. Next slide. And this is an example of how we would take those scores on the left-hand table here and apply it on the big matrix, as you see on the left-hand side of your screen. This is an example of scoring association between CYP2D6 and codeine. It's well known that CYP2D6 is required to metabolize codeine into morphine. So genetic impact on CYP2D6 definitely does impact the efficacy and in some cases toxicity, both for this particular medication of codeine. And so on the left-hand side, you can just see that CPIC guidelines have scored as high, so we're giving a 10 there. For efficacy, it also has toxicity, a lot of evidence for toxicity. It gets 10 there again. So we're applying the scores from the right-hand side to the left-hand side, capping things because there's so much evidence for this. And we get a score of 20, which is our highest. And so that would score a strong, but this association is known to have been repeated over many, many years. So it would be pushed up theoretically to a definitive. It's just an example of how the framework could work. Okay, next slide. Right. So just in summary here, we talked about, why pharmacogenomics is a critical component of genomic medicine. I mean, I think that's obvious that prescribing medication and getting efficacy and toxicity, minimize toxicity, maximizing efficacy is important. And so the framework that I just went over is our proposed framework for gene drug validity. As was mentioned, we're working on one for actionability and for variant classification. And we're hoping to integrate those frameworks into that ClinPGX proposed project that I was talking about earlier, which encompasses PharmGKB, which Lee presented. So I hope you can kind of see in the end how these pieces all fit together. We're going to leverage the stuff that from PharmGKB that Lee showed you, how to curate all that evidence from all those different sources, integrate into these parallel kind of frameworks, and then develop tools and interfaces through our ClinPGX project ultimately. And we're going to be looking for volunteers in the next, hopefully, year or so. So we're glad. Sorry, we're glad to have you guys here today to just kind of like plant that seed in your mind. If you find any of this interesting, when we roll this out, we'd love to have interested curators volunteer. And then, sorry, go ahead, Lee, if you want to go over the last slide.
No worries. So quickly, I just want to list it here in case you want to learn more about pharmacogenomics. So PharmGKB has a page specifically for educational resources related to pharmacogenomics. Please feel free to check it out. So we link to many external education resources for pharmacogenomics as well. So you can learn about pharmacogenomics nomenclature, the tutorials, and direct-to-consumer pharmacogenomics testing, and many of the practice aspect of clinical implementation for pharmacogenomics testing. So, please feel free to check out the resources on this page. And we're really, grateful for the whole GECKO group. The GECKO group is, comprises both PharmGKB and the Stanford ClinGen team. Many of you are very familiar with. And this is led by Dr. Terry Klein. Michelle and Terry are PIs for PharmGKB, and Matt and Terry are PIs for the Stanford ClinGen grants. And we're also grateful for our many members from the ClinGen Pharmacogenomics Working Group. They're from diverse specialties, not only in pharmacogenomics, but we also have experts from regulatory affairs or medical genetics and molecular diagnostics. So I think that's all for our presentations, and we're happy to take any questions that you have. Or feel free to contact any one of us after the meeting as well.
Yeah, thanks for your attention.
So there's a question that's just come into the chat, which I can read out. So dosing decisions in the clinic are based on multiple criteria, including, amongst others, kidney and liver function status, comorbidities, additional medications, medical history, etc. So for some drug dosage decision trees have been created, combining clinical and genomic data and tested in clinical trials. Is this sort of information included in PharmGKB?
Yeah, sure, I can answer that. In short, no, we don't cover everything that pertains to medicine in PharmGKB. We are well aware that clinicians use all kinds of information when they're making decisions about therapies and treatments. Like you said, all different kinds of testing. There's also data that you didn't list, like co-medications, comorbidities, how old is the patient, what are the patient's drug allergies, what other drugs are the patient on, drug-drug interactions. So all of this together, of course, does go into how a clinician prescribes medication or comes up with a therapeutic guidance. And so what we are trying to do is contribute the genetic piece. So in no way is pharmacogenomics a blanket kind of blinders to everything else in medicine, and you just do exactly, something based on the patient's genotype. That's not what we're saying. We're saying we just integrate it in together with all that other information. And that is the job of the clinician. That's what their training is for, is how do they integrate all that information together to come up with the appropriate therapy for the patient.
Oh, there's an interesting follow-up now to that question from Dimitri. So if clinical trials showed that including genetic information doesn't help, would that sort of information then be incorporated into PharmGKB?
Absolutely, 100%. So we curate every, paper that we can get our hands on. Essentially, we're somewhat resource limited. We can't cover all of PubMed, but everything that we can get through, we're annotating it. And that includes things that show that there is no association. As Lee showed with our levels of evidence for our clinical annotations, there was level 4, which is an unsupported category. And what can happen is even maybe a study comes out that shows, like it's a GWAS or something, and they show an association. If that association is not replicated over time, if people try to replicate it and they find, nope, we can't replicate that, that would be knocked down. That association would be knocked down in evidence in PharmGKB. Likewise, that would be the same way with this frameworks. That's why we would have a refuted or disputed category. So yes, absolutely. Information that there's not a clinical relevance for an association might even be valid, but if there's no clinical application, like you're saying, it doesn't help the outcomes of the patient, we use that information. CPIC also publishes guidelines like that, where they go in depth and they see, what the evidence says, no, don't use this in prescribing. Yep.
Just to add on to what Michelle just said, so we annotate both positive and negative studies at PharmGKB, and they will be assigned different scores when we use them for clinical annotation. So the score can go down to even negative if it has a lot of non-association. So this is one part that's slightly different from ClinGen, how they score. Yeah, so our score definitely can go into the negatives. Yeah.
Really interesting to see. I have a question that's sort of more about pharmacogenomics broadly. It's definitely a field that I am not super familiar with. But are there plans either for the future or more immediately of implementing pharmacogenomic testing, sort of like newborn screening is for genetic disorders, as a screening for, or will it always be something that's on an as needs basis?
So that's an interesting question. So there are definitely places that do preemptive pharmacogenomic testing. So that would be sort of like the screening that you're talking about. So for example, at St. Jude's Children's Research Center Hospital, they screen or preemptively test every patient that comes through the door for a full panel of pharmacogenes and looking at specific variants. And then that way it's in the record. And when you go to prescribe medication, you don't have to do a one-off test right then and wait for a result to come back or whatever, which in some cases would impede, impede therapy, right? So that's actually an argument kind of that's used against pharmacogenomics sometimes is like, well, I've got to get this patient on this medication right now. I don't have time to send out for a test. So yeah, if we did some preemptive testing or screening, it would be great. Trying to get that reimbursed right now is a little bit of a barrier. So we are all hoping that one day, yes, everyone could be screened. There's really no stigma. Nobody cares if sitting in your medical record it says you need a different dose of warfarin, right? So there's no kind of really downside to that. People in all the studies that we've seen to date, I don't think I've ever read one where they asked patients if they wanted pharmacogenomic information returned to them and they actually said no, by and large. I'm not saying that there's no patient, but overall there's no studies saying that, oh, by and large patients don't want that information. They do. And so it makes sense to have that sitting in your medical record. Or in Europe, a part of the UPGX project, they get a little card that they can carry around to give to their doctor. And then they can go based on, they can proceed based on that preemptive testing. So yeah, in a perfect world, that's what would be done.
That's super exciting, and hopefully one day we get there. But it seems like there's similar challenges to across the rest of the genetics field, that it all comes back to insurance and reimbursement. And
yes, who's paying is usually the big
that's the barrier.
Yeah.
Well, we are just about at the hour, so we might bring it to an close there. But again, thank you so much to Michelle, Lee, and Clarissa for a great and what was a really comprehensive both introduction to pharmacogenomics as a field and then also to the work of the working group. So thank you so much to the three of you.
Thank you for having
Definitely,
us.
if anyone's interested in volunteering.
I added our
for this
contact
group,
information in the chat. So if there's more questions, feel free to contact us. Great. Thank you.
definitely reach out. Thank you.
Yeah. Bye.
Hey.